Hey, good morning. Welcome back. Starting Unit 3 today. Unit 3 notes. Unit 3 is from DNA to protein. So it's kind of a connecting unit between Unit 2, where we talked about nucleic acids, and Unit 4, where we will talk about protein. So actually 3.1, going to pick up right where we left off. In Unit 2 with mRNA editing. All right, so we're picking up right where we left off. After transcription, that was the last notes we took, transcription, the mRNA gets edited. So during transcription, we had our DNA and we transcribed it into mRNA. We made it a code of mRNA. So let's start off with our mRNA. So there's that again, a single-stranded, looks like half of a ladder because it doesn't have the other side and its rungs. So it's only single-stranded as mRNA. And what we're going to have happen here is we've got these proteins. So these circles I've got drawn, you can kind of see they're pushing the mRNA up. They're making these loops. And these proteins are called spliceosomes. So that's what these proteins are. And they're actually pushing the DNA up, or the mRNA up, excuse me, and then bringing together, at the same time, they kind of bring together these edges. And what they're going to do is they're going to cut here, and here, and here, and here. They're going to put these two pieces together. So you make one long strand, and then you're going to release the parts of the mRNA that gets pushed out. So again, we're cutting that mRNA into several pieces. These pieces here are called introns. And the final piece, the piece that we want actually is called the exon. So well, let's take some notes about these. All right, so important to note, this is before the mRNA leaves the nucleus. This is all happening in the nucleus, and that's where the DNA lives in the eukaryotic cell. That's where transcription occurred, was in the nucleus. So the DNA is transcribed into mRNA. And then before we leave the nucleus, these spliceosomes, again, they kind of push the mRNA up. They cut here and here, here and here, and then they mend together the pieces down at the bottom. So this piece right here gets tied to that piece right there. And that word is splice. That word is splice. When you're putting DNA together, you're splicing it. So it's going to splice together those segments of mRNA and those segments of mRNA, and that's what yields your exon. So the introns, these guys are the worthless or junk RNA. We are not going to end up translating them into protein. They just remain in the nucleus. They get recycled back. There's an enzyme that's going to cut up each of those individual nucleotides and just recycle them. Our cells are actually really, really good at recycling. So that's what our introns are going to do. Our exon, and this is the piece that's been spliced back together. This is the important part of the mRNA. It's now called a mature mRNA. It's ready to be turned into a protein. That process is called translation. It happens in a ribosome, and that's actually where we're going to pick up with our next lesson. We'll pick up right with translation. So a couple quick things to keep in mind. Introns remain in the nucleus. Exons exit the nucleus. So good way to keep track of which is which. The exons exit the nucleus and get turned into protein. The introns remain in the nucleus and are recycled back to nucleotide. So again, we'll pick up with translation next week. Have a great weekend, and we'll see you next time.